without further ado, I welcome you all to this session on countering TR4 in bananas through agroecological innovation. We're very excited to start off, hopefully, and kick off a series of these types of events. Now, just as a personal introduction, so my name is Daniel Szczepański, and I'm a Program and Partnerships Advisor here at AgroCord. So AgroCord is, just to give you a bit of context about how and why we came to this, AgroCord is essentially a network of 13 NGOs, of nonprofits that work globally. So we run about 88 projects in 33 countries across Latin America, Africa, and Asia. So up until the last 5 years, we've actually reached about a little bit over 3 million farmers and over 570 farmers organizations. So our mission, as you can deduce a little bit, is that we work with farmers organizations. We seek to empower them as accountable and effective institutions and as representative bodies to provide effective services to their membership. Now, that is simply what we do ever since we've been created since 2004. We do so, we seek to work with FOs both through project work and knowledge sharing events like this one, based on the challenges and needs that the FOs themselves have identified. And this brings us to the topic of today, countering TR4 in bananas. So to discuss this, we've brought together a panel of distinguished speakers from a variety of organizations. This includes the Alliance Biodiversity International, CIAT, as well as the Australian Centre for International Agricultural Research, as well as one of our farmers organizations from the Philippines, FarmCoop, that we work with. I will leave them to introduce themselves as they start speaking. I myself don't know much about bananas, so I will not take up too much of your time on this. Suffice it to say that today is more about contextualization. It's to provide you a bit of a background about each of the projects and how each of them has seen and tackled this particular issue. So just a couple of small house rules. Please mute your mic during presentations. This is for the audience, or when other speakers are talking. There will be a short Q&A also after each presentation. We can also field some questions right after all of them are finished, but don't worry, we will have time for you. If you do have a question, please raise your hand when you can. We'll try to get to you as soon as possible in the right order. Feel free, though, to use the chat whenever you want to. So that is open to you. I see now, actually, that because we are supposed to have in the program a distinguished speaker from the East African Farmers Federation, but I believe he has been unfortunately delayed. So what I can do in the meantime, while we wait for Stephen Mutiri, the CEO of AF, is that we can do a quick icebreaker quiz, and I will try now to work with my limited technical skills to make this work. So please let me know how it goes. So just give me 1 second. So you should see now appearing on your screen a small quiz. So there's only 3 questions related to bananas. Again, this is just based on my knowledge of bananas, so I hope I did it correctly. Please let me know if you see it. So the questions go essentially, which country currently has the highest banana production? India, China, Brazil, or Colombia? Then which country has the highest banana production per capita? China, India, or, well, Daniel is tricking us. It's totally St. Vincent and the Grenadines. Where, and this is obviously, this should be very, very clear to some participants, at least, I hope. Where is the world's largest collection of banana germplasm located? So that should be a tap-in for some participants, especially. So I'll give you a few minutes just to go through it, also to acquaint yourself a little bit with the controls in the quiz. I see people are answering vigorously, so this is very nice. I'll give you a few minutes while you settle in and drink your coffee.
Lagi?
Okay. I see a very even spread so far between the questions. This is quite interesting. Some people are not falling for my trick questions. If you're just joining us, welcome to this session. I see there's quite a few of you that have just joined in the last few seconds. So I have just started this session, so feel free. We're already running a small quiz, an icebreaker quiz, so feel free to fill in the questions at your leisure. You don't have to. It's not an obligatory quiz. There's no tricks here. It's just for fun. Unfortunately, I have no prizes either. So the only thing you can win here at best is respect. That is the only thing. So I'll close it in 1 more minute. So if you have some answers to change, things that you're considering, please fill in your answers now, or forever hold your silence. Very glad to see also such strong representation from some of our members. So welcome also to us, for example. All right. I'll give you a few more seconds and then I'll close it. Okay. So what are the results? Which country currently has the highest banana production? In fact, is India, actually. So India, apparently, according to the latest statistics, produces about 37 million metric tons. This was in 2024. So quite an astounding volume indeed, actually. India is top. It's mostly for domestic consumption, though. So if you've never heard of Indian bananas, this is very likely the reason. Then, which country has the highest banana production per capita? In fact, this was a bit of a trick question. It is St. Vincent and the Grenadines. So St. Vincent and the Grenadines, they produce about 555 kilograms per person, according to the latest statistics. But of course, it is a very small country. So that answers that. While India, for example, in terms of volume, produces quite a lot, at the same time, they obviously per capita have very many smallholders. So the production per capita is a lot lower. Where is the largest collection of banana germplasm located? As some our colleagues, of course, from Bioversity International, you have no excuses. You should know this. This is in fact in Belgium, in a small town called Leuven, where also my alma mater, the KU Leuven, is located. So they actually host the International Musa Germplasm Center, Transit Center. I believe they contain about, what is it now, 1,700 samples? Is that correct? Yeah, I see people nodding vigorously. So yes, in fact, it is Belgium. So if you ever want to visit Belgium, if only for not beer or for other reasons, then that is also a very good reason if you're interested in bananas. So there you go. So after this icebreaker quiz, I'm unfortunately do not see our colleague from the East African Farmers Federation joining. So without further ado, I would actually ask in that sense that maybe we can just move on to our first presentation, and then we'll see what happens with our colleague. So perhaps we can do this in alphabetical order, if I think that is the Alliance of Biodiversity International SIAT. So Sébastien, feel free to take it away.
All right, I hope you can see my screen.
Yes.
All right. So good morning for me, good afternoon for you all. It's a pleasure to be invited to this seminar series. Indeed, the biggest collection of bananas is in Belgium. I'm located in Belgium. I'm presenting from Belgium, so that's why I said good morning. We are an international CGIAR center, and we have we focus on several crops, as you can see from the slide. You see some rice there, some beans, some cassava, some forages, and of course bananas. We do have also, so agroecology is really the core of our business, I would say. And we also have the gene banks. Of course, no agroecology without agrobiodiversity, right? So we have the gene bank on beans, we have a gene bank on cassava, we have a gene bank on forages, and of course, as said by Daniel, we have a gene bank on bananas. So the gene bank of bananas is in Belgium, the rest is in Colombia. But today, of course, we will focus on bananas and more specifically on Tropical Race 4 and agroecology. So I will give a brief overview of the lessons we have learned and how agroecology and biodiversity can be one of the solutions to combat this disease. Let's see that we can move slides. No. Yes. So some history. I guess you all know how commercial banana production started. It started with Gros Michel arriving in Jamaica a long time ago. And then at that time, it was quite peaceful and quiet in the sense everybody was doing agriculture in a quite nature positive way, I would say. And things went on. Of course, at a certain moment, Gros Michel was the selected variety for exports to the States, to Europe. Big companies started growing. And this is around, let's say, 1900. A little bit before, indeed, the Panama disease was discovered in Panama. Panama disease was discovered, and our friend Gros Michel is unfortunately susceptible, right? And then, of course, at that time, knowledge was not as it is currently, right? And it spread very rapidly, as you can see. It went up. Reports were popping up from several countries that were affected. Also, Africa starts to show up here. And at a certain moment, basically, it was simply impossible to continue this highly intensive banana production monoculture on Gros Michel. Many people think, oh, Gros Michel was devastated, was extincted. Yes, the commercial high intensive production was indeed, of course, the variety itself not, and actually it's still in many countries the preferred variety above Cavendish. But it was simply not possible anymore, that intensive monoculture, and so the whole industry switched to what they had at that time, the less preferred variety, Cavendish. But the Cavendish, of course, was completely resistant to Fusarium, at least to that type of strains, what we nowadays call race 1, right? Race 1 affects, of course, was discovered with Gros Michel and affects actually several other banana varieties, but definitely not Cavendish. And so people thought that was the solution. And then people started high intensive culture of Cavendish until at a certain moment, of course, there was a report of a Fusarium strain that also attacked Cavendish. At that time, it was also not clear. Nowadays, we have genetic tools to sequence those Fusarium strains. And basically, we talk about two different race 4s. that both can, of course, can attack Cavendish varieties, but the subtropical race 4 attacks Cavendish only when there is additional stresses, for instance, cold temperature, and so you mainly see it being a problem in the more subtropical regions. And we can debate whether the one reported at that time in Taiwan was subtropical race 4 or race 4, but it's a fact that not long afterwards, as you can see, around the region, many different reports were popping up, even in the full tropics of Cavendish being heavily affected. And that's basically what we are facing also today. Different races, I will focus a bit on our work in mainly China, Laos, and Vietnam, of course. And what happened in the region, but what people thought about we could contain in Southeast Asia. Asia is of course not true. Not much later, it were reports outside of Asia, Africa, Latin America, quite recently Latin America, and I don't know for those who have been following the recent developments, also the first case in the biggest exporting country was reported in Ecuador. So it's for sure that we should learn from history. Simply replacing Cavendish with another tolerant or resistant variety will not work, right? It's a matter of time before, of course, those microorganisms change much rapidly than we can develop or discover new banana varieties, of course. And so there is more needed than that, and I will explain a little bit of time explaining this. So let's zoom in on what we learned in the region. So a case study of Laos and Vietnam. It was indeed reported. Whether it was already there or not is another question, but the first report popped up in 2018. And also we did some extensive surveys during several years, as you can see. In 2018 is in green, in 2020 is in blue, and then we went to some places, we went twice to see what can we find and how bad is the situation there. At that time, it was still mainly constricted to the northern regions of both countries. There were some cases in the south, but the majority was in the north. At that time, it was limited to Cavendish cultivations and, of course, mainly associated with large-scale plantations. But of course, now 2018, we had quite some genetic knowledge, and we could sequence the genetic diversity of the Fusarium strains, right? And there was quite diversity in there. We found quite some diversity in there, pointing towards multiple entry points and pointing towards that it is quite well established. So meaning that, yeah, the disease entered much before 2018 and much before people were able to study it. At that time, the pathogen was mainly spreading locally and regionally. Of course, you can check that by the analysis of the genetic footprint of the fungus. And of course, how does it spread? First of all, planting materials. As you know, bananas are vegetatively propagated. Even if you don't see symptoms, pathogens can be present, and that's by far the biggest way of spreading diseases. And of course, we should call a halt on that. But of course, also vehicles, machinery, people's shoes are a source of spreading. You might know the fungal disease on the leaves, Black Sigatoka. This one spreads by the wind, right? There is not much you can do to protect your farm, but a soil fungus, and with decent knowledge and intervention and management, basically, in theory, you can keep it out of your farm, and that's of course a very important measure we should recommend to people. But at that time, we saw that from the surveys that the local knowledge of the epidemiology and the disease management was quite limited, and we are still struggling against this. So that's super nice that we have initiatives like this, that we have seminars, so that can people be aware of where to seek help, where to be trained, and how to solve problems, right? We went back actually quite recently. The study is published this year. And we see that since 2018, there was a rapid spread across the most northern provinces along the Red River. But the situation is much more complex, as you can see. It's not only Tropical Race 4, but it's also other Fusarium strains going around. And yeah, we need to be aware of that. So bad news, Fusarium Tropical Race 4 has now spread from mainly monocultures also into smallholders mixed cultivar systems. So if you were hoping I will do my farm in an agroecological way, so in that sense, I can keep the disease out. Yes, you can keep the disease out if you have really nice proper management and good seed systems, healthy planting material, and so on and so on. But if not, unfortunately, we have seen that also the disease enters also small farms. And so, of course, it's not only the export-oriented production that is targeted by the disease, also local production is basically suffering. We know also that, of course, if people grow other varieties, luckily in banana growing countries, there is not only Cavendish, people also like local varieties, which really would like to promote, of course, and one of them is Pisang Awak. Pisang Awak is a very popular banana that was also susceptible to Fusarium Race 1. But we see that in the fields that it's doing quite well versus Tropical Race 4. And another quite popular banana, Pisang Mas, is so far in our surveys remains unaffected. So this is basically good news. If you think that within our banana diversity, there is no variety that is resistant/tolerance against Tropical Race 4. I have good news. There are, right? There are. This doesn't mean, again, please don't go and grow them in monocultures. We really need to think of agroecological production, healthy soils, of which I will explain a little bit further. So basically, those findings indicates that deploying less susceptible local cultivars within diversified production systems is a practical way to sustain banana cultivation. Also, rotations is a very, very smart way to do that. Pathogen pressure is not built up. And so, yeah, having knowledge about disease is key. So surveilling is absolutely key. Check your farms, check your colleagues, check your planting material, invest in seed systems that the planting material you get is really healthy and true to type. Of course, work with resistant or tolerant varieties. And of course, we need to improve our agricultural practices. We should stop growing bananas in a sterile field because, of course, then the disease is spreading. So what basically do we know about the disease? It's a very, very old disease. As you know, wild bananas, crop wild relatives come from Southeast Asia Pacific, right? And we know that there are several wild bananas. And basically those wild bananas, they co-evolved with their pathogen, with their enemy. And so this means within wild banana populations, they acquired resistance, right? And there is still a lot we didn't conserve. If you see this map here, basically, a long time ago, when the seas and the land was different, there was basically a big sea gap here, which we call the Wallace line. And people think that the fungus at that time could not cross this vast amount of water. Here, between the islands, the sea levels were different. There were land bridges. So the fungus developed here in this part of the world, but didn't cross the Wallace line, and so didn't enter New Guinea or Australia. Are there no wild bananas there? Yes, there is 2 wild bananas there, Banksii and Schizocarpa. And you should know that there is no, as far as we know, there is no edible banana without having Banksii as ancestor. So this means that probably that every edible banana has some susceptibility in there. But of course, luckily, they also have other ancestors that were exposed for decades towards the fungus. And so there is resistant mechanisms in there. And so that's why also we started studying. So you see, for instance, here, the genetic composition of Cavendish. And every different color piece of DNA comes from a different wild banana. So we have Malacensis, which of course has a good source of resistance. We have Banksii, which is supposed to be a susceptible wild banana. We have Zebrina, we have Schizocarpa, we have Halabanensis, and there is still an unknown ancestor that maybe is already extincted. So there is still a lot that we need to learn. But of course, knowing the composition of your edible bananas, you can see the resources, possible resources of resistance. And of course, this is where we need to explore our diversity. Luckily, we have also our collection. It has been mentioned several times. And also within our collection, we have resistance and tolerant varieties. This is a list of it. And so this is also freely available for you. So please make use of them. Order them, test them under your own conditions, see how they are behaving, see whether you like them or not, because that's exactly why we have our gene banks. They should not be dusty libraries. They should be books we open. They should be books that are read and are used. But of course, resistant tolerant varieties, I already hinted, is not the sole solution. We also need to adapt our management, right? And for that, we go to China, where we have been experimenting with different intercropping systems and also seeing what is getting there. Of course, availability of seeds for intercrops and suitable intercrops is something we need to investigate. We need to see what different kind of microorganisms they attract and they can keep for that. But we have proven in this publication here that for sure, for instance, this Ciratro cover crop is nicely increasing the soil organic matter, is nicely increasing the nitrogen content because it's a nitrogen fixer. And of course, it's able, and that's maybe the most important one, to attract a stable bacterial community, which we are investigating quite a lot. We are investigating quite a lot in soil health to see which microorganisms are not only, like, for instance, fixing nitrogen, but are also basically combating pathogens. There is a whole list of them. There's whole possibilities. The challenges is basically how to keep those microorganisms stable in our fields, and as one of the solutions against of many, of course, against this terrible disease. We know that in China they are growing basically a combination. It's very popular to have pig manure as a source of microorganisms, and they combine this currently with a resistant/tolerant Cavendish clone, and it seems to be doing quite well for the moment. So that's something we can learn from there. But of course, the best thing is to keep the disease out of your farms. And so we really need to heavily invest in early disease warning systems. This can be, for instance, from satellites. We work with drones, and we combine those drones with an app that everybody of you can download on your phone. It's free to use. So I would say, go to the Google or the Apple or whatever store you have which your operating system is working on, and download it so that you can really learn and help us. For instance, from drones, the software alerts this might be a sick plant, and then of course you need to go to the soil and go there and analyze. So that's how the app looks like. I will play a short video. It basically asks you questions to take pictures of what you suspect is something suspicious, right? And it asks you to take a picture of a certain plant, and it will give you, it will think it's artificial intelligence. Of course, we have trained that, and it will give you a certain answer, and it will talk to you, right? For instance, it says here, it looks like the symptoms corresponds to Xanthomonas wilt of banana. Can you confirm the systems? And then you need to interact with the system. And then he will ask you more questions. You ask you to can you take a picture of the pseudostem? Does it look like this? It interacts with you, and it gives you a diagnosis, possible diagnosis with a certain certainty. As you can see here, he will ask a question, take a picture of the fruits, and of course, once he has this, it comes to a diagnosis, and you can say, no, it's not tropical race 4 in that case. We are looking at the bacterial infection that, of course, is mostly transmitted to tools, maybe also through insects. And we are basically quite proud to say that we almost eradicated this bacterial wilt in Africa. So I invite you, of course, to test this tool. And of course, this only works if we collaborate with local national agricultural research institutes. They need to survey, they need to talk locally, we need to reach out with partners, and we need to train people. So we are training the trainers. People need to get knowledge, And so the first thing is, of course, this is from our experience in Latin America, where we have a big program in containing and detection. So the first point is prevention. Of course, the second point is smart disease management, detect the disease, management disease, keep it contained. Of course, stimulate to grow more tolerant and resistant varieties, because this is part of the solution. But then, of course, you need to see which kind of, not every banana has the same properties, have the same taste, so people should like them, the market should be ready for them, that's for sure. So of course you need to do some socio-economic investigations, and of course coordination, that's absolutely key. So basically my dream is to have a quite diverse set of seed systems, of things that work locally, instead of growing the one banana champion all over the world in an intensive way that has been proven already doesn't solve the solution. So let's work towards bringing more diversity of bananas, more diversity of managing and growing those bananas. And I think, yeah, we will get somewhere. So that would be my short seminar, my short presentation. I have presented the work for sure. I didn't do the work. We were able to do the work thanks to some partners, also some emergency programs in certain countries. And of course, the work in Vietnam, and I guess Guy is here. If you have questions, please feel free to ask. The work in Vietnam was done by Guy Blomme, the work in China by Sijun, the work in Latin America, I need to go one back, the work in Latin America by Jorge, Miguel, and the phenotyping by Michael. Now our new team leader is Ricardo, and of course the genetic work, the analysis of the genetics and the resistance genes is done by our genetics team in Montpellier. You see there, Mathieu. Thanks for the time. Thanks for listening. Happy to answer questions now or during the discussion. Thank you.
Thanks very much, Sebastian, for this excellent overview. Very comprehensive indeed. Congratulations for your work. It seems very, very interesting. So I don't know if there are any particular questions actually from the audience. I have one, but or obviously a few, but I'll wait until maybe potentially somebody has questions. See any hands. In the meantime, while I wait for hands, I do find the app actually quite interesting. So you mentioned it was integrated with drone technology. Is this correct? As well as AI. So this is actually pretty advanced. So have you rolled this out across just Africa or elsewhere as well?
Yeah, so we have it's in many different countries being downloaded. So we are super happy to see that, but in a coordinated way with, let's say, natural agricultural research institutes and farm corporations and so on and so on. The most advanced indeed we are in Africa and in Latin America. So we had a big GIZ project where we were working on Colombia, Ecuador, Peru, Bolivia. So that's where we are really far advanced. I mean, this integration with drones with the app, because of course you need trainings. Drones is, let's say, quite latest technology, and we need to take those in steps. But yeah, also making technology available for day-to-day farmers is really important. So we are also working on a chatbot now, that people can ask questions to the chatbot about their management. And this technology only works once you have experts correcting. So the technology is as good as the database is, of course, right? So we are still further improving that. And yeah, we all can contribute to that. So that's also more of kind of citizen science approach. The more people are sending us pictures, the more we can improve that. And so the coordinated, like for instance now the case in Ecuador, one farm was detected. Of course, you need to scan the area, you need to contain the disease, you need to do the right management, you need to raise awareness, of course. And as I said, don't exchange tools, don't exchange machinery, because that's people how think the disease arrived in Latin America, right? But then, of course, like in Peru, irrigation. In Peru, we have a very good production of organic bananas. It's in a dry area where the leaf fungus has no effect, let's say, but of course you need to irrigate a lot. And there, of course, the pathogen enters the river and the water system. And then of course, it spreads very, very rapidly also through water and irrigation. So we need to be aware of that and yeah, be a step ahead, right? And rotates. We are now also studying how long the disease can survive and on which wild plants it can survive. So we have still make very big progress to make, and we need to combine all the solutions we have.
Okay, excellent. Thank you. A comprehensive answer, yeah. Okay, well, I don't see any other questions in chat or anywhere else. Can you talk more about wild bananas? I see a question from one of our audience. I don't know if you can mention that a little bit.
Yeah. So wild bananas was for a long time a big gap in the sense when we started the collection more than 35 years ago, we focused on edible bananas. We wanted to collect as much as edible bananas as possible because, of course, breeding is quite challenging because edible bananas are sterile. And so if you see towards the breeding programs, the usage of wild relatives is quite limited. Let's say only 5 individuals have been used in of that we are aware of in a breeding program. So this is definitely not far enough because we want multiple angles of resistance, not a single gene resistance, and not a gene for gene. Yes, this works, but this will be broken in a very short time, right? So we are working on that. And wild bananas for a long time have not been accessible and neglected. So we have now, in collaboration with Mesa Botanic Garden, started a seed collection. So we have went out on a mission of to collect wild bananas, study the population diversity in there. So we started the seed collection. You can also order seeds from us now. And of course, we are studying those because there is quite some diversity within those wild bananas. Very interesting to know our history, of course. And we have seen also there is multiple sources of resistance. If you see the current somaclonal variants that are on the market, they have different mechanisms of being tolerant / resistant, right? And so that's what we need to build on with, let's say, classical breeding and modern breeding. And luckily, we have been, I see that some people from Vietnam are here. We are collecting in Vietnam. We were collecting in Papua New Guinea. We were collecting in Australia. Well, for Fusarium resistance, maybe that's not the hot spot to find resistance, but of course for other traits for sure, yes. And so yeah, we are really happy to collaborate with other countries to see what we can do to find multiple entries, multiple alleles that can contribute resistance and tolerance.
Okay, thank you very much. I suggest that we move on to the next presentation. So if you have more questions also for Sebastian, feel free to write them in the chat or to simply ask later. There will be time. So thank you very much, Sebastian, for this excellent presentation. And we move on to our Australian friends. Tony, you're up.
Okay, I'll unmute myself now. You can hear me? And,
yes. Yeah.
And share my screen. You can now see that?
Yes, we can. Yeah, thank you. Go ahead.
Great. I love it when it all works. So a big thank you to everyone for allowing me to speak today. I work for the Queensland Department of Primary Industries, and we are funded by the Australian Centre for International Agricultural Research, or ACR, to do work. And for the past 20 years, we've been doing work in Southeast Asia on Fusarium Wilt of bananas. And it's through these experiences of visiting farms that I've been able to get some insights onto what has happened on banana growing areas. So I'm going to give a very quick overview of our latest project, which has been running for 6 years now. And just Sebastian, thank you very much for giving a great overview of TR4 and the problems that it's caused in the region. And as you know, it's caused by a fungus, Fusarium oxysporum specialis cubensis, which is a soil-borne fungus, and it's I guess what's been classed as a forever disease. So once you've got it, you can never get rid of it. And as Sebastian showed very nicely, it started in the Indo-Malay area in the 1990s and has spread throughout Southeast Asia. And I had 26 countries. I now have to update that to 27 countries. Thanks, Sebastian. I wasn't aware that it had been found in Ecuador. So the project that I'm talking about is, well, it's been based in Southeast Asia around 4 different banana producing countries, and they've been selected purposely because because they have different banana production systems, commercial large scale Cavendish like in Queensland, or a smallholder commercial Cavendish in the Philippines. We've also seen Lao, as Sebastian talked about, has a mixed type of farming systems where there are large commercial ones and smallholders growing pisang awak. And Indonesia is a really interesting case because it has so many small farmers and diversity of different varieties. And that's probably where we've learned that you can grow susceptible varieties in a system where it is suppressive. So through the early years of their ACR project work, we were able to see growers successfully growing these susceptible grow Michel type with TR4 all around them. But but they were able to then also able to grow successfully and with minimal losses. So it was really understanding why was this happening. And throughout this project, we've also managed to have collaborations with Malaysia, Uganda, and South Africa. But we've focused on smallholder growers as the main producers who have limited resources and limited technical knowledge. So the project was designed to have 3 interconnecting objectives, and we tried to build it from the ground up, literally. So understanding the soil organisms and the role they play, because it's the microbes in the soil that are the first defense against Panama TR4. They're the ones that are surrounding the root system where the disease enters the plant. So if we can understand what the microbes are doing there and how they can provide these healthy plants, we can get a better understanding of how we may be able to manage that disease and manipulate that microbiome. The second objective is around management. So we're using management then as a lever to manipulate that microbiome. How we can do different, how we can use different management techniques then to enhance or suppress different microorganisms. And we're looking at the different crop rotations, cover crops, and also then what does that do in economics. And the third objective, and equally as important as they're all interacting into one another, is the engagement with the banana growers, having the conversations with the growers so that we're learning about what works on their farms as much as we're giving information about disease management. So I'm going to go through these in a little bit more detail, but not a lot of detail just because of the time constraints. So the first objective, as I said, was about the microbiome research. And I've really broken this down into 2 parts. The first one was about understanding the specific microorganisms, those specific core organisms that give that protection to the banana plants. And through our work with our collaborating partners, we were able to establish that an international core microbiome, a group of organisms that are found in healthy plants, no matter whether the bananas are grown in Australia, Philippines, Indonesia, Malaysia, Laos, Uganda, or South Africa. And that consisted of 74 bacteria and 59 fungi. And 2 of those specifically we've studied in detail because these give us the greatest suppression of TR4 in our assays. And that's what I've tried to describe very badly in the graphic below. It really requires a whole webinar presentation just on this topic to understand how those organisms work. But they're able to build better biocontrols by understanding that. And it also gives us a lead into how the general microbial activity, things like how does land use affect soil organisms. So within our region in North Queensland, where the bananas are grown, we've found that where bananas have been cultivated, we've halved the microbial biomass and diversity compared to native areas. So where those wild bananas that Sebastian was talked about in the rainforest, where we find those, we've got half the biomass. And particularly, we've really depleted the fungal component of that microbial biomass. So what we found is that where we have the biomass, we also double the likelihood that TR4, Fusarium oxysporum cabensi, can establish in those soil. So that's quite an important finding, is if we have the biomass, we double the likelihood of disease. And similarly, if we go the other way around, if we double the biomass, we can then halve the likelihood of TR4 being established. So that microbial manipulation becomes a very important factor. Just moving on to the second objective, which was about farm management strategies. And I wanted to start off here by focusing on the inherent risks. These are some of the background risks that run just by growing bananas. And one of the big ones is your location of your farm compared to an infected farm. And this is something that a lot of smallholder growers get hung up on. Not banana growers everywhere around the world that I've met. They're very focused on what their neighbour's doing or not doing. But we want to change that to look at the induced risks, those management practices that growers can concentrate on, where they can actually do something about it. And this comes down to 5 main disease management principles: prevention, the biosecurities we talked about; protection, which is really bioprotection, increasing those the microbes in the soil to produce a healthy plant; reduction, this is reducing the amount of disease inoculum in the soil. And if we're going to get these other strategies to work, this is just as important. We can't have high inoculum soils and try and manage the disease. Resistance, which Sebastian spoke about, and we find there's limitations there, that most markets are already established on well-defined varieties, and it's the ones that are most susceptible that give the greatest price. So growers tend to revert back to what they know and try and grow those high-value susceptible crops. And there's very much is the commitment that the banana growers and banana growing communities show to disease management. So understanding how we can commit to sharing that knowledge and exchanging that knowledge. So just a bit more detail about some of the protection strategies. And I've broken this down into 2 main areas of how we can attack this. One is the in-crop. And again, this is about growing diverse vegetation, having those ground covers. And we've shown that where we increase the amount of ground cover in and around banana plants, we can also gain a lot more organisms that are suppressing Fusarium in the soil. So we get 66% of the organisms of soils that are 66% of organisms interacting with Fusarium actually suppress it in that one with a high ground cover, where in a bare soil, we only get 1 organism, or about 10% of the organisms interacting with Fusarium. Nutrient management plays a big part in the susceptibility of banana plants. And I'm going to talk about this in a bit more detail, but it seems in commercial banana production, where we're growing monoculture Cavendish, Williams grand name, 400 kilograms, more than 400 kilograms of nitrogen per hectare seems to be the tipping point. We need to come back from that and get smarter nutrient management. And the same with phosphorus. The other side is the out-of-crop management. So what we do between banana crops to reset the seed. And using organic amendments, like high carbon amendments, to really stimulate that fungal biomass and get it back in the soil. Fusarium isn't a good competitor against other fungi. And this is probably why we see so many different types of trichoderma actually being quite successfully used. Fusarium doesn't like fungal competition. But also, there comes a point between banana crops where we need to reset that system. And we've looked at different cash crops versus green manures, but there needs to be a period of 18 to 24 months at least between banana crops to bring that inoculum level down to a level that it can be managed in the preceding banana crop. The other part of this management strategy is destruction. As I talked about, it's inoculum management, and it's been part of the success in Queensland is being able to pick up those plants really early. If you can pick it up with a drone, it's too late. Because the disease does the old leaves first, it comes from the bottom. And as soon as those leaves go yellow, you need to take that infected pseudostem out, get it bagged, removed out of the plant system so it doesn't return the chlamydospores that build up within the infected pseudostem back in the soil to build that inoculum up. And that's what we've been doing through the Queensland system, which now has only 9 farms that are infected over 10 years, which has been quite a successful program. So it's not perfect, but it has been working well for us in our system. And we've got a lot of feedback from stakeholders just running some workshops just recently, and all the growers that are on those infected farms fully support the destruction protocol. Even though it costs them around $2,500 for each zone, they see that as an investment in the rest of their farms. They're quite large farms. But the success really depends on that timely pseudostem destruction, removing the plants as quick as possible, using urea, particularly with that pseudostem material, bagging it up and putting urea in with the bag so that you get this fumigation of from the ammonia that's generated, and that also helps to break down that material, so making it less likely to carry disease over. There needs to be a period of soil recovery, and we need to have an adaptive method for the climatic conditions, and it needs to be evidence-based. And that's what the growers have told us that they want from a destruction sites, and they're quite happy to support that. So the third one is about engagement. Third objective is trying to keep banana growers engaged, and there's a lot of barriers to that and limitations. And so we understand that not all growers have autonomy over their farms and the decisions, so we had to try and work around that, and then also provide a sense of hope that this disease is management. And within that engagement, we did a lot of work with the Fall Armyworm Action Plan, the ASEAN Fall Armyworm Action Plan group, about the role women play, the important role women play as IPM leaders for management of TR4. And on the screen there is a link to that site where the videos are of the different countries talking about the important role. We also need to focus on the opportunities and solutions that are most practical to each group of growers and the individual farmers. And to understand the impact of our interventions, for example, some of our disease intervention strategies have shown under business as usual scenarios, the growers can expect plant losses of 71% to TR4 quite quickly. But under modified farming practices, this came down to 17%. And similarly, over 5 cropping cycles, we were able to show that we could turn a $14,000 loss into a $41,000 profit by using smarter management practices. But we also need to understand the message that we're delivering about TR4 management. If we continue to focus on biosecurity, which has been successful in Australia and Queensland, we're probably only likely to get a 4% increase in uptake of new practices, and that's over an 11-year period. But if we include agroecology and how we can manage the farm practices to suppress the disease, we're likely to get a much higher rate, around that 94% adoption over the time. So the messaging is as important as the message. Now I'd like to move on to a decision to support tool that brings all this information together that we've developed over the last 20 years from our ACR funded research. And it starts by recognising not all farmers face the same disease pressure. TR4 can be no threat for some growers, and then it can be a threat, then it can be an incursion on some farms and widespread on others. How banana farmers manage the disease is different in each of these situations, and we can assess the farm management practices and use the 6 disease management principles I talked about earlier to develop a unique farm disease management risk to make better decisions. And I'll show you how this works very, very shortly. And it's an Excel-based tool, and the tool is best done with a facilitator by having a conversation and exchanging information. So it's not a one-way delivery of knowledge, it's an exchange of information as part of the process. So firstly, we select the scenario by answering 2 questions. The first question is the disease on your farm or not? Depending on the response, the grower will be prompted a different second question. If it's not on the farm, is it in the region? If it is on the farm, how widespread is it? These questions can set how risk adverse or accepting a grower is to TR4. So based on those responses, you're directed to a different scenario. In this case, we're using scenario 3. And then once you select your scenario, you're prompted to a different worksheet where you work through a set of questions. And then, oops, sorry, I'll just go back. I'll just get this to the point where I wanted it. You get a different set of questions. So we have our disease risk outcome score here, but we also have the likelihood of different outcomes. Firstly, there's a likely incursion of the disease onto your farm if you don't have the disease. The next is what's the likelihood of the disease spreading on your farm? In this case, it seems to be quite low at 33%. And then is what's the likelihood of the farmer losing their whole crop and their farm? Then as we work on further, then we look at the different individual prevention management practices as we see at the top of this graphic. So we then can, in this example, prevention and disease reduction practices are low risk. Protection and people are moderate risk, and resistance is high risk. And we need to understand why our resistant practices are so high, and are we willing to accept that, then work through it. We can then use then define where we put our resources, into which part of the management of the disease are we going to get the biggest impact. So some of the impacts that our project has, we've really got a greater understanding of how soil microbes interact with the banana plant to protect it. We're trying to get this shift into how the disease is management, more into ecological disease management, being proactive to prevent the disease rather than reactive once you have the disease. We're trying to give growers decision support, and through these the Fusarium risk tool that with some scenario based and really tailored to individual farms. And we've already shown that we can, through good farm management practices, we can turn around that $14,000 loss into a $40,000 profit. And we've included a better understanding of how we can include women as part of that role in understanding and delivering that message. So some of the lessons we've learned is about smarter agriculture rather than increased inputs is what's important. The value of an integrated approach. There's no single silver bullet solution to TR4. The more different practices you can include into the management of the disease, the better the impact. We need to understand that social science and why people do what they do is just as important as the biophysical. And quite importantly is the partnerships, making sure that we're listening to the people in the country and working with them as we develop up our research capacity in each of our partner countries. So the future, I think, is really through that ecological disease management. And quickly, it's a mix of precision input of organic and synthetic fertilizers. It's vegetated ground cover to increase that microbial diversity, about inoculum management, removing those infected plants before they have a chance to build up that high disease pressure and changing the landscape of farms so we can produce banana islands instead of having a sea of banana monoculture. It's about how we get better biocontrols through biological enhancement, getting that grower engagement and providing the tools and exchanging information and that knowledge sharing. So just to wrap up in the conclusions, we've really defined the importance of the soil microorganisms over the past 6 years within this ACR project. We've shown the practical applications, both in crop and out of crop, how that can be used to manipulate the microbes in the soil, and we've given greater grower empowerment. And as I've said before, we can turn that around from a loss into a benefit, into a profit. And it's really about the long-term banana production requires that agroecological approach that Sebastian was talking about. So through continued collaboration, innovation, knowledge sharing, we can provide better livelihoods for banana growers, no matter the size of their farms or where they're growing bananas. So with that, thank you very much. And thank you to the Australian Centre for International Centre for Agricultural Research. And thank you to everyone for listening and for AgriCord for inviting me to be part of the webinar, and to the big team that sits behind this project in all the different countries and my collaborators. So with that, I'll stop sharing and hand back to you, Daniel.
Yeah, well done. Yeah, very, very impressive indeed. It was very interesting to also hear you speak about ecological disease management, but also these other aspects about smallholder engagement. I mean, this is obviously music to our ears. And the aspect about livelihood support and engagement, I mean, this is also, of course, critical. And the fact that you were able to actually turn this around into a profit per hectare is extremely vital. As of course, you know, this affects livelihood. So I don't know if there are any questions so far for Tony. Feel free to put it either in chat or just raise your hand. I do have some questions with regards to the tool, or at least one, because I mean that seems also quite interesting. I'll wait maybe a little bit to see in case anybody. We have some questions also for Sebastian, but I see that they have been answered already. Can return to those later. But yeah, with regards to the tool, this is actually quite interesting. So it's an Excel-based tool, correct? So you work directly with farmers organizations to actually roll this tool out with farmers. So
yeah. Sorry, it went through a bit of a very much an iterative process. So it started off as a paper-based tool, understanding what people wanted to talk about, what were the important aspects, very much based around biosecurity and some of the farm management components. But as we were talking to growers, people management became a very important part of that, having that shared responsibility of the disease on the big farms, so that everyone was invested. So through that, it's developed through a number of other iterations, and we've taken it over to our partners overseas and worked through what was important. And this seemed to be the best way to capture a lot of information quite succinctly. So I'm hoping the tool will be publicly available by the end of October. We're just waiting for some publications that support the use of the tool to be finalised, and that should all be. The deadlines are end of October, might be middle of November, if depending on how everything goes. But certainly by the middle of November, it will be available. It's it runs a few macros just to help with navigation, but the idea is also in the future to sit down. It's that facilitation and that knowledge sharing, which is equally as important as a printout of the outcomes for the growers.
Yeah, thanks. I see quite a few questions actually in the chat popping up now. So what I'll do is I'll turn to the first one, and then for the others I'll move it later because Khalil has to also talk in her next presentation. So don't worry, I'll come to your questions at some point. The first question, though, for you is if you could elaborate a bit more about turning losses into profit. Of course, this would be one of the questions. So you've mentioned this number quite a few times, particularly also from the smallholders' perspective. If you could elaborate on that.
Yep. Yeah, for sure. Thank you. That's yeah, an interesting question. It was one that came out of one of our research trials in the Philippines, where growers, where the trial was on a university farm. We were using what they call Tall Williams as the variety, so it had a little bit of resistance, and we did inocular management. But what we were looking at was the use of the recommended rates of fertilizers, the standard practices of fertilizer, which was like 6 weekly applications of a large amount. And then we changed that to still using the same amount of fertilizer, but split the applications and then also used it as a solution. So with that, we were able to also reduce disease, but also pick up productivity. So over that time, we didn't get the same disease pressure as we did under the standard banana production, standard fertilizer regime, and that's where we got a large amount of our losses. So we I think the end of that trial, end of the 5 crop cycle, we're close to 71% mortality within the plants. Whereas by the end of that same period, where we had better nutrient management, we were much better. Where we could even get better reductions in disease losses if we used biological sources, but unfortunately they were also less productive because the nutrients weren't available as readily. So I think as Sebastian might have already mentioned, it's a mix of how do we use synthetic fertilizers with biological fertilizers so that we have the nutrients available for the plants to use, but at a much lower, steadier release, so we're not getting those big ups and downs and imbalances in the microbial community, which is what seems to be being affected.
All right, interesting. So a mix of smart nutrient management, the 5 crop cycle leads to, or at least in the trial you've had, to an increase in income. Simplistically put, yes.
Yeah.
Yeah.
Yes. Yeah.
Okay. Well, that's interesting. I'm sure that some of the people in here would like to hear more about that. But we do have a bit of a time pressure, so I now kindly thank you, Tony, for this excellent presentation. We'll come back to some of these questions more for you later as well, Tony. But now we have to move on towards the Philippines and our colleague, actually, our AgriCord colleague, Khalil. So Khalil, please feel free. You have the floor.
Thank you, Daniel. And thanks so much, Tony and Sebastian, for your presentations. It seems like there's some overlap in our work. Daniel, the only thing is we have some videos, right? I'll try to see between the presentation and because I'll insert the videos in between, so I'll go back and forth, hopefully. Okay, hold on.
Yeah, no worries. I mean, we have a bit of time, so it'll be okay. Please let us know if there's something we can do.
All right, I'll be going in and out, so hopefully it's not too disruptive. Let me see. Just a minute. Okay. Hi everyone. There's just some videos that I'll be showing in between my presentation. Okay. So, hi, I'm Khalil Ito, and can you see this? Is that clear?
Yes, we can see your screen. Yeah, go ahead.
Good. I'm Khalil Ito, and I'm with the Foundation for Agrarian Reform Cooperatives in Mindanao. I'm a soil scientist and agroecology practitioner, and I lead the agroecology research and development unit, as well as the Land Rights Research Consortium of Farm Co-op. So our banana cooperative partners, CFAR BEMCO, DARCO, and MDS ARCO will be presenting with me the farmer organizations leading research and innovation for the project, which is aimed to co-develop local agroecological solutions to tackle Fusarium oxysporum cubensi, folk TR4. So first, I just want to thank AgriCord, Trias, We Affect, and Thousand Currents for the support for this work. And just a little bit about Farm Co-op. We are a grassroots non-governmental organization supporting farm cooperatives and also associations with memberships of over 6,000 farmers who grow bananas, cacao, coffee, and coconuts under monocrop and multi-cropping systems. Of the 22 banana farm cooperatives we work with, we've partnered with DARBCO, CFAR BEMCO, MDS ARCO, and DARMOPCO for this project. However, because of land rights issues, that's affecting all of our cooperatives under the banana ARB system, we've lost DARMOPCO as a research partner during this time. But a little bit on the experience of our DARBCO, CFAR BEMCO, MDS ARCO cooperatives. They were established in mid-1990s, and most of their members own about roughly 1 hectare each, and they export outside of the country and have inherited the monocrop banana system. And because of the fair market price that Farm Co-op has helped negotiate with the cooperatives, their income has increased a great deal since the inception of their cooperatives. However, over the years that the folk TR4 has infected their farms, production has reduced, and so has the income of the co-ops. So the capacity of the cooperatives vary in terms of being able to be part of the research, but they're very interested in finding local solutions. So we're very honored, and also we find it meaningful to be working with them directly on this. So all of the cooperatives, the banana cooperatives we're working with have been affected by folk TR4, except for the organic indigenous areas we're working with. And just really quickly, we have a 40 team working with the cooperatives in the field, but also creating compost and also organic inputs, vermicompost, and also sourcing indigenous plants and seeds from the indigenous community that is part of Farm Co-op's network and partnerships. And we're also incredibly grateful for the support and constellation of partners, agroecology researchers, practitioners, and farmers and indigenous CSOs who've been part of our learning in this process, and whose meaningful work has informed ours and has also strengthened our process. So why is this important? I think Tony and Sebastian already covered that. So according to FAO, 400 million rely on bananas as staple food. And of the 135 banana producing countries, 23 countries, or probably more now, worldwide are affected by folk TR4. And in the Philippines, 15,000 hectares are infected by folk TR4. And of those 15,000, 11,000 are owned by smallholder farmers. And we work with 4,000 smallholder farmers who are affected by folk TR4. And in the Mindanao study, 57% of 372 farmers who were surveyed experience at least 50% decrease in income. And I believe that study also said that about 43% did not receive government support. Oh, so now we asked our banana cooperatives about their observations of the farms prior to the Foray project. So let me put that video in. Hold on.
Before siya naabot, ang Fusarium na nagyud sa area. Distributed din sa area. Walay area diri nga walay Fusarium. Dismayado na mi asa naman mi anik. Unya na yung mga nangabot nga mga kanangkuan, abot asa na sila gikan mo promote og product. Kami kay kuan mi mo palit pud mi pero maghihapo epekto. Ang yuta kan kumpak. Gahi ng yuta. Wala ni tanom. Di na mo tumuha kay mo na dito nangahurot halos ang ganina hurot tanan ang kuan niya. Ang saging nito pita.
Sa una gyud makuan ako ang sagingan walay sagbot kay ginapatay yun ang sagbot na yung budget ang planter sa pangpatay sa sagbot. Abono mo man na among makita gyud kay naaman chuitik abono na yung nga mao gyud na iyang work diha sa field lay ang mag-abono lay ang mag-spray sa sagbot. So ang sagingan is dili siya nindot tan-awon kung naay green ang ubos.
1970s nga time mo itong pagsugod na sa saging, maayo kayo sa umpisa. Maayo kayo ang appearance sa mga saging tungod kay ang yuta manggod atong panahon na fertile pa kaayo. Daghan kayo ang gigamit na mga fertilizer, synthetic fertilizer para makaproduce o daghan kaayo nga produkto. Nabalakan nami nga ang yuta, gahi na kayo sa dugay na kayong paggamit sa yuta nga nagagamit mga synthetic. Na-notice gina mo na gahi kayo ang yuta ang imuhang mga gan.
Tungo na sir, init dito ang lugar sir. Nya baho ang area. Wala kayo sa saging.
1971 po rin. Gwapo saging. So isa po nga nakakuan sa yuta noon kay nagkaanam siya og kakumpak. Mo na problema karon. Dili ang among production ko dili ko parehas na nagkaanam siya og kakumpak. Mas laborious ang conventional kay ang sagbot nimo dapat ma-maintain nimo. Pero kung organic imong natira o mga compost, dili mapud ko nga kinahanglan gyud kada bulan mo tira kay kada bulan kinaami sa permanente god.
Sa una batuon yun na sir niya murag na portion diha nga gagmay og saging tungod kay kumpak ang yuta diha murag gahi ba. Ang tagiya na sa una na gamit yun siya og synthetic fertilizer.
Naa naman gyud ko na tao diri no na witness gyud nako kung unsa gyud ang itsura o sinugdanan sa amo ang lugar no. So dako siya kausaban. In terms of soil, makita nimo ang soil sa una o soil karon na gyud ginatawag na depletion of nutrients yun. Kita gyud nimo didto nga mura nagkagamay ang lawas sa saging. So naagyoy kausaban.
Kanang nabutangan sa trial, solid yun na siya nga Panama. Infected yun na siya, hurot yun na siya diha. Kana nga area is kumpak ang yuta. So di halos madutlan o kanang shubel ko imong anaon palahon.
Okay. Well, it's a very interesting video. Yeah. I don't know if you want to go ahead with your presentation.
Sure. Hold on. Okay, so this fits, sorry about that long delay, but this fits right into one of the topic we were talking about, interventions. According to the dialogues we've had with the cooperative farmers, there has been some interventions in the past from government and research institutions, the universities, and also companies. But the results have been variable. especially on introducing resistant varieties. Some of it worked, but still there's been some spread of TR4 in some of the areas. The introduction of trichoderma was also had variable results. And then there's entrepreneurs who would come bringing in their biocontrols, and that can have disastrous or so-so results. And so there has been some distrust or some hesitation regarding experimenting around this, although the farmers have been doing their own experimentations as well. And some have been so desperate that they also use things that should not be used for the plants. So it's been a very interesting conversation. When we started talking about doing a farmer-led research, we had to be very sensitive to the concerns of the farmers in terms of containing this work so that we could actually work on the process before it gets scaled. So in our agroecology research, we integrate multiple ways of knowing. Traditional knowledge especially is incredibly important when we're trying to find out locally adapted plants, but also experience empirical knowledge of the farmers and indigenous folks on the different kinds of plants that are available and what they've seen in terms of the interactions of these plants with other plants. And those are hardly really in literature and have yet to really be uncovered. And it's been an honor for us to be working with these plants and to bring them back, actually. So we also, of course, integrate institutional research, and some of them are studies from different countries that has been informative. And then we also, guiding our work has been the principles of agroecology, indigenous principles, as well as principles around soil health, biodiversity, ecosystem restoration, and land regeneration. So through participatory action research, we did establish 3 trial areas, 4 originally until we lost one of the cooperatives because of the land rights issue. And we had 2 criteria in addition to others that are typical for site selection. One of them is that there needs to have a history of TR4 in that area. And if we could limit or have no resistant variety in the areas, just so that we can see whether these practices are actually working and we can respond right away and change the practices. So we needed something really susceptible and also very sensitive to the practices. And then we brought in soil microbes through the compost, vermicompost, and organic inputs that we were experimenting with, and then cover crops, companion planting, hopefully to optimize plant-plant interaction, soil-plant interaction, and other symbiosis that we hope would come through to support the diversity of soil microbiology that we're trying to promote in the area. In addition to that, we had to do some biosecurity, containment measures, and then continually reevaluate the risks and work on the adaptive management that we're doing for each different area, trial area that we're working with. So we did have control, but the variability was so great amongst the different cooperatives and in the landscape, and the farm situation. So those controls were not really working. But we have some results that are very useful to the cooperatives. So now is the time to bring in the second video.
Pagka after pila ka bulan, siguro mo 3 months, makita na namo ang soil, nanay kalahiin na yun, humok na siya ba? Naon humok na siya. Anya ang matanong mo na dito, vigorous na siya, green na siya. Compare mo sa palibot, green din siya.
Pag-abot sa agroecology, kinahanglan ay sila as covered crops ang magturok sa ubos. Na ay infected nga, gikan siya sa infected mother, follower siya, na-harvestan siya.
Karon nga na kuan na siya sa agroecology, sigihan na mo siya og apply og compost.
After na gigamitan na mo aning agroecology farming, aduna na din siya kausaban. Nag-change na ang appearance from yellowish to naging green farm na siya. Tako siya ka-usaban. Of course, makita ni mo una ang dahon sa saging, nagbaga, and nag-green. And also parang mga iyang mga bunga. From perting kagmaya, ni din siya pag-usab ni dako na ang iyang bunga.
Maayo ang iyang epekto when it comes to plant health, sa iyang vigor, and then sa mga diseases sa tanom. Effective gid siya, kay wala naman kaayo yung mga sakit dito.
Moisture niya, retention niya na. Kaya nang hamog, bugnaw siya. At the same time, kuhan ko lang yung bugnaw niya. Mag-retain na niya siya moisture. Hindi na yun siya dali nga mauga.
Here comes with the agroecology introduced to us. Dako kayo may pasalamat. And within these 3 years, dako jud changes ang nahitabo. Unang-una, ang soil nga dati compacted. It was tested, no? Natin mga photograph videos dito. Pero natingala ka nga ang sa itsura pa lang daan sa soil, visually makita nimo nga naa nag-changes. Kay from clayish nga brown nga buak-buak karon medyo nag-darken siya ug nag-soft na nga yuta, no? Pero ang pinaka-amaze nako nga nakatingalahan within the 3 years ang pagpatay og mga plant diseases like FOC nga 15 to 20 cases per week. Imagine for 2 years wala zero siya ba? Zero ang panghitabo. So it really naka-amaze sa ako nga nanagyo pa day pag-asa nga with this agroecology na pwede di ay nga dili na mabalikan og fusarium ang area.
Nagkagamay ang infestation sa ginatawag mong milibag. Sige na mo siya apply murag kuan siya baka nang murag repellent na bitaw siya sa mga ubang kuan except tong mga beneficial insects na naa dito. Sa dahon pud niya makita na jud nimo na nag-expand jud ang kadakon siyang leaves. Tapos green na siya.
So we thank everyone who's been part of our journey, especially our cooperatives and also our funders and so many others in the community. Thank you.
Thank you, Khalil. Thanks for this presentation. I don't know if there are any questions for Khalil, actually, from the audience. There are some, actually, I believe, in the chat as well that I can address. I also have one question sent in beforehand that I could post to the panel and to everybody else. So one of them relates to actually, I believe, intercropping as a strategy to counter TR4. So I don't know how Sebastian, Tony, and Khalil, how you have approached this topic. Has intercropping worked as an effective management strategy to counter TR4? That was by one farmer from the Philippines, apparently.
I can go ahead, and it has worked in our cases. Not our experience isn't so much with TR4, but with race 1, where we have seen declines in the amount of Fusarium infected plants with cover crops present. So we're very much aware that growers need to produce bananas. So I look at what's growing around under the bananas as weeds and cover crops, and they're different because cover crops are a managed system, and for us in our systems it's a lot about selecting what's a low growing species that allows workers to move in amongst the banana plants to continue their work unimpeded, but allow those microbes to continue. And there are some very good low-growing, shade-tolerant species that get that build that will help with that suppression and build that microbial diversity. And it was great to see that from Khalil's videos, just how diverse that is in the Philippines, because we have been having troubles getting introduced into our collaborators.
Yeah, also from our side, farmers in general might be a bit reluctant because they're not used to it. So for many of them, it's new, right? But as Tony said, indeed, we have plenty of experience with race one. If you surround susceptible varieties with plants that are not a host for Fusarium, for sure, it really helps. For us, it's also a matter of, again, there with cover crops, there is no magic bullet for all. Very important is availability of seeds. They should be readily available for local farmers. They should do well in local environments. Guy can also give a bit more experience from our side in Africa, because again, what works in Africa does not necessarily works in the Philippines as well. Of course, also the general soil health is super important. If you have high nitrogen levels in your soil, of course, those beneficial microorganisms as nitrogen fixing bacteria or mycorrhizal fungi, they're quite sensitive to that, and this will not work. So people need to be willing to really adapt their complete farm management, be open to that. And of course, soil health is super crucial. So what we see, as I showed from our experience in China, yes, indeed, some microorganisms are really competing or even fighting Fusaria strains. But of course, it's a matter of keeping stable microbial communities, and this is also sometimes quite a challenge. We often see that. For instance, in the lab work, it really helps, it really works. And then after weeks in the field, competition with other local microorganisms plays in, and those beneficial fighting microorganisms are also competed out. So we are really learning soil structures, climates, moisture content. This all plays a role, and there's definitely not one solution you can roll out, let's say, across the world. So you really need to learn what locally works, what is locally available, and you can really set up also a business on the cover crops. You could see if it's not too vigorous, if they don't start to climb, can they be used to harvest, to feed to cattle, things like that. So you really need to work together with the cooperatives and the local people and develop local solutions.
Okay, thank you. We have some more questions actually, but I believe Khalil, there has been one directed at you. Have you tried using thermal compost with cover crops? And I believe you've answered it. So I don't know if you want to add to that.
Yeah, I just saw it more clearly. Like, have you tried using thermal compost with the cover crops? Yes, but we don't have enough cover crops as mentioned. That the problem in the Philippines is there's really not enough of the seeds, and that's what we're trying to bring back. So we're trying to get as much cover crop and materials in there, but it's not sufficient. We're working on it though. But in terms of cover cropping all over the field or just around the trees, we're trying to make it all over the field. And so that's why the agroecology trial is a bit tricky when you actually start having that percentage of coverage in the area. So there's variability for that. I think that's a challenge in the Philippines, finding enough seeds. And the best place we were able to get more is actually from the indigenous communities, not from not from the universities, unfortunately. So it's definitely a need, and I hope there will be a growing source and supply for that. But the other problem is actually labor for harvesting the seeds. And we've been working on cover cropping since I think 2019 for quite some time. And it's not easy to grow and find the right ones. And again, it's something that we're still watching to see which ones would work well. And we found something that works really well with bananas, but there's not enough source. I hope I answered your question. No.
Yes, thanks for feeling that. I believe there is a follow-up question I meant for compost application, if all over the field or not. Yeah, I don't know if you have any further comments on that. Yeah.
It's around, there's some in the areas, but there's around the bananas, but also we're trying to see whether we could do some broadcast. We're also working on extracts and such because there's going to be a supply issue with compost. But we're using the soil food web approach for the compost and actually what was mentioned earlier on wood chips by Tony is something that we're actually improving. And that's another issue is getting the right wood chips in there. And, there's a time, it's also a temporal consideration that we need to have with wood chips, especially in the tropics where we don't know whether those wood chips are, you need to let them compost a bit before using it. So there's a lot of those variabilities that we're trying to figure out and finding the right trees. And there's so much more to talk about when it comes to wood chips. I can go on about it. And it's been great to be talking with folks in the soil food about it too.
All right, interesting. Yeah. Noor, thanks, Noor, for the question. She seems happy now. Thank you very much. So I'm not sure if I've covered all of the questions. I think there are some more. If I've missed them out, please feel free also to just raise your hand and discuss a bit more. I think there's one more for Tony, actually, that I've missed from Mohammed Rahidul Islam, I believe. Please explain how you expand the collaboration on integrated management in other countries. I'm not sure if you can elaborate a little bit more on that, Tony. For regards to IPM, yeah.
Yeah, for sure. I think with the tool that we've got now, it makes a lot of sense to for expansion into other countries that aren't there. So we're not starting from a zero base as we have done before. We can work through, well, what are the issues for your growers in your situation and move forward and build on the knowledge that we currently have. As I said earlier, my funding comes through the Australian Centre for International Agricultural Research, and so they're quite open to funding. But they have target countries because we're based in the Southeast Asia Pacific region. So obviously Southeast Asia Pacific is our main target. But having said that, there are banana projects also in East Africa in some of the collaborating countries. So the best way is to get in contact with me if you want to get in with collaboration, or through ACR for the Australian Centre for International Agricultural Research, or the national bodies that for banana growers that are represented and have a say on what are the important issues that they fund.
All right. Very interesting. Thank you very much. I think there was one new question here. Can we use M. balbisiana, M. acuminata to be parent for breeding TR4 resistant strands? I think this might be a question for you, Sebastian.
Yeah. We didn't find too much sources in balbisiana. Like I said, we did find in the several acuminata subspecies. So yes, this is definitely a way to go, and especially Malacensis and Burmanicoides are quite well known for their sources of resistances. So yes, and there are still others that we didn't explore too much. And like for instance, Halabanensis is quite rare and only a few, also the Burmanica, only a few individuals are in collections. So we really don't know population differences, but that's exactly what we are going to do, and trying to organize also more collection missions and together with the local partners, study their diversity, conserve their diversity. Also, we are starting to conserve pollen with the specific aim indeed for breeding, that you are one step ahead. But that's all things that are being developed together with partners.
Interesting. Thank you very much. Yeah, I don't know if I've missed any questions. There's still quite a few in chat, so please feel free to make any comments as you wish, because we are drawing to a close now. What I will say, though, is that there are very many interesting also complementarities between the relevant expertise and tools that we've all developed. I can imagine the complementarity between, for example, the Tumaini app that the Biodiversity International has pioneered, but as well as your tool, Tony. I think I can see very interesting complementarities and lessons to be learned across. So I think this could be also an interesting discussion point for the future. And I think this is also something that Khalil asked about, even whether there had been even coordination between or any possible coordination for future action. So I think this is something that could be interesting to explore, perhaps moving on towards the future. Yeah, for the audience, thanks for coming today. We still have a few minutes left, so if you have any questions at this point, please feel free to raise them. I do have one final poll actually for you. Don't worry, it's not a very long one. It's more related to discussion topics moving on towards the future. If you're interested in doing one, so let me just see. Feel free to ask any questions while I go through. So you should see now a new poll appearing on your screen for questions. So would you be interested in learning more about TR4 and how to manage it? The second question would be, would you be interested in another webinar? And if so, what topic do you think we should focus on? I actually implemented a few myself, but yeah, feel free to put in whatever you like. There's a fourth question, what do you think we should talk about? So there I left it more open. So feel free to also put in some items if you feel like my focus has been a bit more reductionist, to put it this way. Yeah, okay. Thank you for filling it in. I see already quite a few answers. So far, quite a few people want to continue the conversation. That's very heartening. It would have been very awkward if people suddenly said no. This is actually a good development. All right. I do think there would also be, so far, digital tools is ahead together with prevention and risk reduction. Those are two interesting topics that I think people would be interested in exploring more. Obviously, this is also a hot topic, digital tools, smart technology. I think this is quite something that's interesting to a lot of not just local partners, but also financing partners. We're seeing that also more and more from our side. Low costs, but accessible tech is something that people are interested in exploring. So far, that one is definitely up there. But yeah, quite a few answers for all of the above. So prevention, risk reduction, pest management strategies, digital tools, all of them have received actually quite a bit of interest. So I'll wait for a few more minutes. Feel free to ask any questions you may still have at any point. But so far, I believe it's pretty unanimous that we should, in fact, continue the series. So what we can do actually also afterwards, together with the panelists, is that we can have a separate session with you afterwards, and we can discuss both the outcomes of the poll, but we can also see, based on the interactions here, how we can move forward. I very much hope that you're still available and willing to continue this path. I mean, from our side, there is no problem whatsoever. In fact, we'd be glad to host more webinars. So yeah, that's just a short input from my side. As said, I'm not an expert on this matter, but Agricord is likes to view itself as a broker when it comes to these types of knowledge sharing events. And so we're very happy to put the experts together and see what we can come up with on specific topics. So we do hope that this is indeed the start of a conversation. So I'll wait a few more seconds and then I'll just end the poll. glad to see also quite a few farmers and not just us basically talking to ourselves. So that was also quite nice to see. All right, I'll end the poll. So thank you very much for your inputs. So I will now just to share the results with you. So it's pretty unanimous. Everybody wants to learn more about TR4. Everybody would also be interested in another webinar. What topics to cover? Essentially, I think people said pretty much unanimously, well, not unanimously, but the highest amount of answers did indicate all of the above, especially on prevention and risk reduction. But there's a nice spread. At least nobody said none of the above, so that's actually quite nice to see as well. I think there were also some other answers, so I will try to see what people filled in the other questions. So just give me a second, please. I will share also, obviously, as said, all right. I will share also with you the videos that Khalil has sent. And what I would suggest actually is that together with the panelists, we have a short meeting during the week if you're available. And then we can also discuss the outcome, as I said, of the poll and of this. So unless anybody has any more questions, we're actually out of time. I don't have the possibility to see what people have filled in on the last question, but I will try to digest this, and I will forward you the replies. But I think there are quite a few people that have actually suggested alternate topics. We'll go through with those together with the panelists. We'd like to thank you all on behalf of AgriCord for attending this session. I think it was a very interesting exchange. I hope we continue this. And as said, Sebastian, Tony, Khalil, I hope we can meet also during the week and continue the conversation. We were very happy to host you all here, and we'll share the recording with you afterwards. And again, thank you very much for attending this session. Special thanks to the panelists. Great job. Excellent presentations, and let's continue this. We're seeing lots of love being given. That's very nice to see. Thank you very much, and have a great day. Thank you. Yeah.
Thanks.
Thanks very much.
Thank you.
Yeah, thanks.
Thank you all. Hope to see you in the next meeting.
Yes, likewise.
Thank you.
Thank you.